Good afternoon. My name is Sara Wrobel, and I'm the European Product Manager for ACD and Genomics at Bio-Techne. And I'm very excited to be talking to you today at Spatial Biology Europe Online. With the rapid growth of the use of spatial genomics technologies, users are now able to gain new insight and answer previously challenging questions. In this presentation, I'll be showing you the expanding toolbox of RNAscope technology as a multi-omic spatial analysis discovery and validation tool. Even in the not too distant past, understanding gene expression when you're looking at the techniques that are available to you, there are only really two methods that you could look and two paths down which you could travel. Using molecular biology methods such as PCR or next generation sequencing, using that molecular detection to understand the expression of genes within complex tissues. Obviously, with this becomes challenges, potentially complicated workflows, and more importantly, the inability to differentiate the origin of sequence and the lack of cellular resolution. On the other hand, going from the gene directly to the output of the gene, the protein. And for this, or even the RNA, using IHC or traditional in situ hybridization techniques. These broadly based histochemical methods are great because they look at the ultimate, sometimes the ultimate gene output, but they are reliant on the availability of good tools, for example, good antibodies, for often where antibodies are not an option to visualize gene expression. Potentially they suffer from a lack of sensitivity and specificity, and essentially lack of standardization often of protocols. And this is into a field where 10 years ago now RNAscope stepped in, joining the sensitivity of the methods of molecular detection into giving that spatial overlook with other methods such as IHC and FISH. Essentially bringing both the best of both worlds into the toolbox for researchers. But why? Why is understanding the cellular and morphological context of gene expression important? Well, because highly heterogeneous tissues such as neural tissue, the brain, the tumor microenvironment, and understanding organoids and developmental models are comprised of multiple cell types and states. And by understanding the precise transcriptomic characterization of these tissues could enable identification of new cell types, predictive biomarkers, and new therapeutic strategies. Only through interrogation of these complex heterogeneous tissues can we understand them and characterize them. And the tools that are needed to do that often needed to be highly sensitive, very specific to the genes that you're looking for, and often multiplexed to give a truly spatial approach with single cell resolution. From the inception of tools of next generation sequencing, then came an evolution of transcriptomic tools, from bulk transcriptomics, very bind and grind tools to allow precise gene expression analysis, but without any single cell or spatial information. Then came the development of single cell transcriptomics, which really took the understanding of gene expression at a very high plexing level to the next step, being able to understand the gene expression in a single cell, but again, missing that spatial context. Much more recently, there's been an increase in techniques available within the spatial transcriptomic field. And many of these techniques allow that very, very high level of plexing to understand the transcriptomic environment within a piece of tissue, but also have started to be able to introduce some spatial visualization. And alongside all of these methods, the ability to understand gene and analyze gene expression in tissue and cells with a single cell resolution and spatial orientation sits alongside all of these as a technique which, applied with all of these, really gives a full picture and an understanding of gene expression. And why are we here today? Why is understanding spatially resolved transcriptomics so important? Well, you're probably all aware that in 2020, and published earlier this year, Nature Methods published that spatially resolved transcriptomics was one of the most important areas and applications in research around, that spatially resolved transcriptomics are essentially changing the way we understand complex tissues. And at ACD and with our RNAscope technology, we're really proud that we've been at the forefront of spatial transcriptomics for over 10 years. Since RNAscope technology was released onto the market, our first publication came in 2011. And since then, we've had over 3,500 peer-reviewed papers published in numerous journals, including many top-tier ones. In 2020 alone, as with the adoption of spatial transcriptomic technologies, we had a 30% jump in our publications. We found through working with researchers worldwide that our technology is highly relevant across multiple fields of research, with over 40% of our publications in cancer research, followed then by a high level of publications in neuroscience and infectious diseases. And furthermore, the incorporation of RNAscope technology and that visualization of spatial genomic information at a single cell level has been rapidly adopted by people wanting to understand what's going on with their and spatially map and confirm their single cell RNA-seq data. So as the techniques of single cell RNA sequencing has grown, so has the people joining both of our technologies within the tissue context, with over 80% of these single cell papers that are using both single cell RNA-seq and RNAscope technologies in very high impact journals worldwide. So before I go into some applications and examples of how RNAscope is being incorporated in a wide range of research and at various aspects and solving various problems for people trying to understand the spatially resolved gene expression, I'd first like to go into a little bit about the technology itself. If you're unfamiliar with RNAscope, it's an RNA in situ hybridization method that obviously allows RNA detection in tissue and cells with morphological context, giving both a single cell and a single molecule resolution. It consists of three parts. The first here on the left is the target probe, and this is a probe that is designed against your sequence of interest to be highly specific. The second part is the signal amplification system, and then this generates a really high signal to noise ratio. And lastly, the visualization of the single RNA molecules as dots. Now, the assay allows for spatial mapping of a variety of different RNA species. So that could be messenger RNA, long non-coding RNA, and through the expansion of our technology platforms, we've been able to stretch this all the way down to various mRNA species down to microRNAs and very short RNA sequences. All of these can be visualized dependent on the assay with either a fluorescent or chromogenic labels, which really gives end users the flexibility. We also have the ability and support the running of our assays on both some automated systems and to be able to do it manually. And most importantly, the assays can be performed at a wide variety of different sample types, including FFPE tissues, fresh and fixed frozen tissues, PBMCs, and cultured cells. To give you an idea of the different assays that we have for spatial mapping of gene expression, we currently have three distinct technologies, all based around the basic RNAscope double Z technology, which is based and ensures that we have the specificity to our particular targets. We have RNAscope, which allows the detection of RNA targets of over greater 300 nucleotides in length, providing single molecule and single cell detection. And there are a number of different assay options for our RNAscope platform, going from both in chromogenic and also fluorescent detection, going from a single plex fluorescence all the way up to a 12 and now potentially a 48 plex. For researchers who are interested in looking at shorter RNA sequences, looking at discrimination potentially of splice variants, identification of short highly homologous sequence, and even point mutations, we have a technology called BaseScope, which again is based on a double Z probe design strategy. This again also delivers single molecule detection and is available both automated and manually in a chromogenic format. And then finally, we have microRNAscope. This is one of the newest innovations in our technology, which actually has a completely new probe design strategy, and allows RNA detection at a single cell resolution. And importantly, miRNAscope allows the detection of much smaller sequences of RNA. So everything from 17 to 50 nucleotides in length. And this obviously includes miRNAs, siRNAs, multiple other small RNA species that are around in the cellular environment, as well as other more novel molecules like antisense oligonucleotides, which are increasingly used in cell and gene therapy applications. So with this expanded and ever-expanding capability to understand what's going on in terms of gene expression, what are our main applications? Well, as you can imagine, if you're able to target through specific probing any gene expression, the world is your oyster. Particularly, we know that customers really benefit from our ability to look at targets which traditional histochemical methods can't actually visualize. For example, targets which are very challenging to detect by using antibodies, such as non-coding RNAs, GPCRs, and secreted proteins. They also benefit from the ability to multiplex and get that high level of resolution and sensitivity with understanding the expression of their cell markers, as well as having the capability of doing IHC as well as RNAscope at the same time, and the general information and additional information you can gain through the spatial mapping of single cell data. This has been adopted widely in areas which deal with complex tissues in basic science sphere, looking at neuroscience, stem cells, and developmental biology. Within infectious diseases, our ability to generate a probe based on a gene sequence allows us to be incredibly reactive, and which has been widely adopted within researchers within infectious disease. In particular, we're incredibly proud of our ability to support researchers worldwide who've been investigating the SARS-CoV-2 epidemic. Within biopharma discovery and development, we support a variety of different companies and research groups who are understanding their cell and gene therapies, both understanding the models that they're using to understand the mechanisms of how these work, but also assisting in safety and toxicity studies and preclinical modeling to understand where potentially highly gene modified or gene modified cells are within tissues, and as well as understanding and visualizing new innovative RNA therapies. And finally, within we have multiple applications within cancer and clinical research, understanding those complex environments around cancer and oncology. So the tumor biology itself, the microenvironment, the interface between the immune system and the cancer and the pathology of the disease. As illustrated in some of my previous slides, there are many techniques that are being used to understand gene expression, and each have their place in the appropriate phase of a piece of research. Increasingly, a standard approach is not to just use one. It's not a either or method, but to use multiple techniques to get the best possible data and information about gene expression across complex tissues. Be that in discovery, using bulk or single cell transcriptomic techniques, and then further discovery with validation with a single cell spatial analysis, or using many of these new very high plexing spatial transcriptomic tools in the discovery field, and then going on for further exploration and validation with single cell spatial analysis. Each piece of the information is added with these techniques and bring immense amounts of insights to the understanding of complex tissues and the cell-to-cell interactions which govern function. And in the next few slides, I'd like to show how RNA-scope single-cell spatial analysis has been incorporated into multiple steps and applications in spatial workflows. The first place I'd like to start is to illustrate how RNA-scope is able to expand the capabilities and application of the nanostring geomic digital spatial profiler. Working with nanostring to create a workflow, our ACD have been combining geomics DSP with RNA-scope technology. And here RNA-scope can be used to target genes that users feel are important within the selection of regions of interest prior to the higher plexing RNA profiling with the geomics spatial profiler. Also, RNA-scope can be used to validate and further explore the results obtained by the DSP's RNA output, gaining spatial determination of key genes of interest across the whole tissue section at a single cell level. To illustrate how RNA-scope can be used to select the regions of interest, here is a piece of lung cancer tissue that's been stained with RNA-scope to identify the regions of immune infiltration. You can see here they've been selected based on the two RNA-scope markers we've been using, so CD19 for the B cell infiltration and CD3 epsilon for T cell infiltration. We profiled around 24 regions of interest, and on the right you can see the expression makeup of the selected regions. So this varies in predominantly pink, those areas rich in B cells, because they're showing a strong CD9 expression. And then as you work down the regions of interest, you can see other areas that are more green, which indicate higher T cell levels indicated by the CD epsilon expression. And also towards the bottom, you can see more general tumor regions with relatively low expression for both, for a low immune cell content for both types of immune infiltrates. Next, the actual digital spatial profiling of those areas that have been selected within the piece of tissue. And then in this case, this study just looked at 78 different genes to understand the gene expression in these regions. Of the 24 regions of interest, which you can see along the bottom, and those were profiled and selected based on the CD19 and CD epsilon enrichment. You can see that these regions are clustered together based on the gene expression in a pretty unbiased way. And if you look at some of these, the genes in these clusters, you're getting gene profiles that you would expect for the respective T cell and B cell compartments. This giving a huge amount of confidence that we're profiling appropriately with the DSP, and that was enabled by the RNA-scope assay. These regions of interest obviously show those areas that are enriched, and they correlate with the regions of interest enriched for B cells or T cells, but also the gene expression profiles of the regions of interest that showed poor immune infiltration are confirmed by the low expression of these immune cell marker genes. To further explore the expression analysis patterns identified with the panels, we used a multiplex RNA-scope assay to validate and spatially determine the expression of some more key genes across the whole tissue section and down to the single cell level. We performed RNA-scope on 4 immunoregulatory markers, ICOS, PD1, CTLA4, and PDL1. And if you have any knowledge about the tumor microenvironment and the immune system around potentially cancers, you'll know that these are immune regulators, potentially negative regulators of immune response. The RNA-scope demonstrated a very high expression of all 4 markers in the CD3-rich region of interest 11 compared to region of interest 6. This is what we'd expected because region of interest 11 is predominantly the T-cell-rich region. And although area 11 does demonstrate a very strong expression of these genes, region of interest 6 also shows some basal level of expression. Interestingly, in the top right panel shows a magnified image of region 11, where you can really appreciate the single cell resolution by RNA scope. The blue arrow indicated a cell that was positive for PD1 and CTLA4, potentially showing the ability of the technique to identify single cells with potentially exhausted phenotypes, as indicated by these markers. An important take-home message from this slide is that the correlation of the DSP data with the RNA scope staining really shows that these 2 techniques are incredibly can create some great synergies in detecting, understanding, exploring, and further refining what's going on within gene expression. To find out more about the workflow in a white paper that we put together between ACD and NanoString can be found on our website, acdbio.com. But there are many, many other techniques coming or existingly available to researchers that people are harnessing the wide range of spatial tools available to them. The spatial organization of the brain, as many of you will know, is fundamentally related to its function. And the structure-function relationship is especially apparent in the context of the human cerebral cortex, where cells residing in different cortical layers show different gene expression patterns and exhibit different patterns of morphology, physiology, and connectivity. In this particular study, the authors using spatial transcriptomic methods, they identified different layers enriched areas and identified genes that had been previously identified and implicated in Alzheimer's disease and schizophrenia. And these spatial gene expression patterns have really been used to help and potentially may help to refine the organizational context of these different neuropsychiatric disorders and may provide new targets for molecular interrogation. But importantly here, the authors performed single molecule FISH in the scope of RNA scope to validate their data from all of the wealth of data that they generated from a sequencing-based spatial transcriptomic approach technique, as they considered it the gold standard for quantifying gene expression with a high resolution. In another example, an RNA-scope technology has also been applied to study cancer immune cell communication across a whole tumor section without the limiting implications of potentially tissue dissociation. In this particular study, the limitations of the authors identified a current limitation of experimental methods for their planned experiments, because by using traditional IHC, the number of cells or the number of ligand receptor interactions that these researchers were particularly interested in really limited what they could do and the cellular resolution that they could understand gene expression at. So this group of University of Queensland researchers took a really multi-strategy approach to develop a really powerful and experimental analytical pipeline, allowing for the genome-wide discovery and targeted validation of understanding the genes expressed in cellular communication. They profiled thousands of genes and using spatial transcriptomic technique and single-cell RNA sequencing. So they first generated the data to show that of the genes that were possibly involved in the cancer immune cell communication. And then the expression of these candidate genes were then visualized with single molecule RNA-scope and droplet digital PCR. And then they also could develop a computational pipeline that allowed for the modeling of cell-cell interactions using RNA-scope data to recapitulate an interaction landscape across that whole tissue. Now, one of the ongoing requirements for many research is the ability to understand multiple gene expression profiles whilst retaining the single cell and, if required, single molecule resolution. In the study I just described, they did just this using our RNA-scope high-plex assay. It's an application of RNA-scope technology that allows the visualization of up to 12 genes, and with a newly released reagent workflow, now up to 48 targets. For those unfamiliar with the RNA-scope high-plex assay, it allows the high-plexing of up to 12 different RNA targets, enabled by employing cleavable fluorophores and iterative detection. The fluorophore cleaving procedure is rapid, and it has minimal or no effect on RNA and tissue morphology. And the images from the various rounds of imaging can be merged together using RNA-scope high-plex image registration software. And additionally, with our new high-plex reagent workflow, we can enable users now to detect up to 48 targets on a single section. We used this reagent workflow and employed it recently in an internal study to investigate the diverse expression patterns of a range of neuronal markers in the mouse brain. In addition to visualizing and confirming previously described single cell RNA sequencing data, we also focused on 4 distinct areas of the mouse brain to further explore the use of the high-plex as a tool to understanding the regional specific functional significance of these neuronal genes. And this is just one example of one of the areas that we were profiling, looking particularly at the hypothalamus. As you may know, the hypothalamus has a key role in hormone release, regulating body temperature and maintaining daily physiological cycles. And in this particular slide, we visualized LHX6 expressing neurons in a particular region of the hypothalamus. Now, LHX6 has been shown to be really important in sleep promotion, and the loss of function in LH6 has also been shown to really disrupt sleep homeostasis. If you'd like to find out more aspects of the data and the wider data set generated here and the other applications of high-plex, those can be found also on our website at acdbio.com. Not only are researchers worldwide demanding the ability to perform single-cell RNA scope with multiple markers, but though in terms of the number of markers that they're looking at, but also that applies to the gene outputs that they're wanting as well. So to extend and why they're doing this is to increase the understanding of the cellular dynamics and phenotypic expression in heterogeneous tissues. Researchers are increasingly moving towards a multi-omic approach. We've recently enhanced our capacity to support this and support researchers who look at their RNA targets alongside their chosen protein markers with our new co-detection workflow. Well, previous success with IHC antibodies in conjunction with ISH was not always possible. Our new co-detection workflow and reagents enable more antibodies to be compatible with co-detection, whilst reducing the time needed for optimization and antibody validation, whilst maintaining IHC staining and sensitivity. I'd like to finish there, but to sum up, I hope that I've been able to illustrate to you that RNA scope technology is a sensitive and specific solution for spatial multi-omic analysis. And as a provider of single cell spatial analysis technology for over 10 years, we have a wide range of solutions in a huge number of applications. So thank you very much for your time today. And if you have any questions, I would be very happy to answer them.